All right, are there any questions from last time? So towards the end of the last lecture, we were talking about how to distinguish one chromosome from the other by staining it with certain dyes, and then you get what is called a karyotype. Now, if, if you think of a cell, right, it has something like a life cycle, okay? Basically divide, grow, okay? Cell will grow, then it will divide, right? Again it will grow, then again it will divide and so on, okay? So that's actually called a cell cycle, which we will be encountering in more detail in a later chapter. But this cell cycle can be broadly divided into two phases. One is called interphase and the other is called mitosis, right? Mitosis is the division of the nucleus of the cell, right? Now, so there'll be cell division, right, followed by growth, right? Then again cell division and so on. Okay, so whenever the cell is dividing, you're calling it mitosis, and the period in between each cell division, that is referred to as interphase. Now, during interphase, transcription, translation, and DNA replication have to take place because the cell is growing means, the contents have to be duplicated, right? And so the chromosome is in an extended state, and it cannot be easily distinguished under a light microscope. And these chromosomes are called interphase chromosomes, right? It's not compacted. During cell division or mitosis or nuclear division, the chromosomes have to replicate and one copy needs to go to each daughter cell, right? And in order to make the separation easier, the chromosomes are going to be in a compacted state, right? So during this phase, that is mitosis, the chromosomes are highly compacted and are visible under a light microscope, and these chromosomes are called mitotic chromosomes. In fact, chromosomes were discovered when watching cell division, right? In maybe 1800s, right? Under the microscope, they could see the condensation of the chromosomes. So you have interphase and you have mitosis. So interphase means, the chromosomes are not compacted. They're probably being actively transcribed, right? Okay? Or there's DNA replication or whatever is taking place, right? And during mitosis, the nuclear division takes place. So the chromosomes, yeah, go ahead. No, yeah, chromosome will get duplicated, yes, yeah. No, see, the cell contains the chromosomes, all right? It has many other things. So every time the cell divides, all right, all the stuff inside has to be duplicated too, right? So DNA replication will make sure that you can replicate the DNA, right? And then the chromosomes will get duplicated. But if it's like a long string, that is all messed up, then, separating it out will be difficult. So it'll be like compact things, and then the cell division machinery will pull those towards two sides of the dividing cell. So. And we will talk more about this because there is a chapter on cell division that is coming up. Right? Maybe 3-4 more chapters later, we will talk about cell division, cancer, and so on. Okay. So we will look at this in more detail. But for now, just keep in mind that there are two different possible states of chromosomes. One is it's an interphase chromosome, right? The other one is the mitotic chromosome, right? So mitotic chromosome means it's highly condensed. It's very compact. Okay, it's not like loosely lying around. Whereas in the interphase chromosome will be pretty loose because transcription, right? Then DNA replication and so on have to take place. So the DNA in the chromosome has got to be accessible. Okay, so it cannot be like compactly packaged then. Now, chromosomes have got certain specialized DNA sequences to ensure that they replicate efficiently and are correctly apportioned between the two daughter cells during cell division. Whenever the cell divides, right, if you're looking at a human cell, there are 3 billion of those letters on each chromosome. Well, not on each chromosome, distributed among 23 chromosomes. But when the cell divides, the chromosomes have been replicated, and then each cell has to get one copy of the chromosome, right? Properly, it has to be partitioned. If it's not partitioned properly, how many of you are familiar with Down syndrome? Do you know anybody that has Down syndrome? Or you may have heard of this disease, all right? That's a person, when they were the egg and the sperm before fusing, the partitioning did not take place properly, okay? So they got an extra copy of one chromosome. So we have 2 copies of each chromosome. Those guys have an extra copy. They have 3 copies of chromosome number 21, okay? That's why the scientific term for that is trisomy 21. They have 3 copies, and you can see, most cases if your chromosomal number changes, you're not going to make it, okay? We're not going to be seeing, nobody will see you around, but Down syndrome is an exception, okay, where, you still see, they look abnormal, but, you still see people like that walking around, functioning, more or less normally, but most cases, the fertilized egg is not even viable. So to ensure that chromosomes replicate efficiently and are correctly apportioned between the two daughter cells, you have what are called replication regions. We already talked about that. That is regions where it's usually rich in AT pairs because AT has smaller number of hydrogen bonds, alright? So the DNA replication begins at a replication origin. Most eukaryotic chromosomes contain many replication origins to ensure that the entire chromosome can be replicated rapidly because the eukaryotic chromosomes, they are much longer, okay? Then there is also another specialized region which is at the center of the chromosome. It's called a centromere. The presence of this sequence will allow one copy of each duplicated chromosome to be pulled into each daughter cell when a cell divides. Right? So when the cell divides, the chromosome will replicate, but they're still attached in the middle. Right? Then something has to come and bind on one side. Okay, pick up one of the daughter chromosomes. On the other side, the other one will bind, and then they'll pull them apart. Right? We will talk about the details of how that happens. Right? But that specialized region where something can come and bind in order to separate out the two daughter chromosomes, that is called a centromere. Right? So during mitosis, a protein complex that is called a kinetochore forms at the centromere, and it attaches the duplicate chromosomes to what is called a mitotic spindle. Again, I mean, this is the structure that separates the two of them out. We will talk about that in a later chapter. And so the centromere basically facilitates separation of the two daughter chromosomes. And the third region is what is called a telomere, right? This is a specialized DNA sequence which is found at each of the two ends of a chromosome. If you have a linear chromosome molecule, linear DNA molecule ending in a chromosome, towards the end you have this special region that is called a telomere, right? Telomeres contain repeated nucleotide sequences which will enable the ends of chromosomes to be replicated. Remember, DNA polymerase always needs a primer, okay? You cannot start a DNA thing from scratch, okay? Because of that, if you have a linear molecule, right, there's a problem at the end, right? You have to start it with a primer, then you have to worry about Okazaki fragments and things like that on the lagging strand, right? Well, this telomere will make sure that there is some redundancy at the end, okay? So it really doesn't matter what happens, okay? The stuff that really needs to be copied faithfully, that is bracketed in between. Now this telomere, as the cell grows older, with, as the number of cell divisions increases, the telomere gets shortened, okay? So that after a certain point, that cell will not divide. In some of the cancers, there is an enzyme that is called telomerase, okay, that will add those copies towards the end. So if telomerase is hyperactive, that's another, feature that can be found in many cancers, because if telomerase is hyperactive, then the telomere doesn't shorten, okay, and the cell kind of can become immortal, right? That's what, so, and again, do a Google search in telomerase, you will see all of this stuff, right? Not for this course, but just for your interest, own interest. Now, how is DNA packaged inside eukaryotic cells? I hope the bus wasn't late, okay? Is the bus just, no, okay. Is what, in the center? Oh, okay. Yeah, no, I'm not questioning why you came in late, but I just thought, I hope it's not because of the bus. Because somebody complained the other day the bus is not coming on time and things like that. Now, so if you're looking at DNA, how is it packaged? First of all, you have 8 histone proteins, like 1, 2, 3, 4, and then 4 underneath, okay? So that's like the, bead on which you're going to wind a string, right? So then the double-stranded DNA is wound on this to produce this kind of beads on a string appearance. So around this you have 146 nucleotide pairs, right? That is bound on a single bead. And in between these two beads, you have this linker DNA that's about 50 base pairs long, right? So this arrangement, right? This arrangement is going to reduce the length of the original DNA by a factor of 3. Okay, it'll become 1/3. And then there is a lot more packaging that goes in because, to give you some concrete numbers, this is like, taking a string that is like 12 miles long, right? And putting it together and putting it inside a tennis ball, okay? And it cannot be a jumbled mess because later on you're going to need the DNA for, DNA replication, for producing the right proteins and all that, So nature has its way of doing the packaging, and it's very efficiently done using these histones, because histones, these are proteins that have a large number of positively charged amino acids, okay? Because DNA has got negative charge because there's a sugar phosphate backbone. Remember, the phosphate carries negative charges. So regardless of the sequence, all right, the histones are able to attach onto the DNA quite well, right? So the histone octamer forms a protein core. So octamer, there are 8 of these pieces, 1, 2, 3, 4, and then 5, 6, 7, 8. around which the double-stranded DNA helix winds. The term nucleosome refers to basically a nucleosome core particle plus an adjacent DNA linker, which is about 50 nucleotide base pairs long. So the formation of nucleosomes, as I said, converts the DNA molecule into a chromatin thread, which is approximately a third of its initial length. Histones are small proteins with a high proportion of positively charged amino acids. Because of the positive charges, the histones can bind tightly to the negatively charged sugar phosphate backbone of DNA, regardless of the precise nucleotide sequence. Right? And there are additional levels of chromatin packing. This is just one level that we discussed. Some others are conjecture, some have been figured out, right? But the bottom line is that it is packaged really, really well. Right? Now, if you look at the chromatin in an interphase chromosome, again, remember, we talked about interphase, we talked about mitosis. Mitosis is when the actual nuclear division takes place, when the chromosome is in a highly compacted state. So from one mitosis to another, that is the interphase. Okay, so during interphase, it's not that tightly packaged. Okay, but again, even during interphase, the chromosome, all right, the chromatin in the chromosome is not in the same packing state throughout the chromosome. Right, in some places, the packing might be more compact, not as much as the mitotic chromosome. All right, but there will be variations in the degree of packing. Right, heterochromatin is the most highly condensed form of interphase chromatin. Right. And heterochromatin typically makes up about 10% of an interphase chromosome, and in mammalian chromosomes, it is typically concentrated around the centromere and telomere regions. Remember, the centromere and telomere, they have no function as far as transcription, DNA replication is concerned, right? Because the telomere is basically to make more copies. Okay, it adds extra length to the linear DNA molecule. The centromere is the region where the kinetochore is going to come and attach to facilitate the separation of the two the two daughter chromosomes during cell division, right? So it doesn't have any transcriptional or any DNA replication type of function, right? So it is quite compact, yeah. Or it does not produce mRNA, no. It is not for that. Okay, that DNA thing is so that something can come and attach onto it. If you're looking at the centromere, telomere is just extra copies made at the end so that it's like a buffer that has been put in. Okay, so that when you're copying, you just copy the relevant portion. Okay, it's like towards the end. So that's why the heterochromatin can be in a very, very compact stage of packing, right? And it is transcriptionally inactive, right? It is not going to produce any messenger RNA or anything like that, right? Now, one very good example of heterochromatin, right, packing is in the X chromosomes of female mammals, right? Recall that males have one X chromosome and a Y chromosome. Females have two X chromosomes, right, in every cell. But if you have two X chromosomes, then the two X chromosomes will produce double dose of that corresponding protein, right? So that will be disastrous, right? So because of that, female mammals will permanently inactivate one of the two X chromosomes by condensing it into heterochromatin very early in embryonic development, right? Very, very early in life, it is decided, okay, whether it is the paternal X chromosome or the maternal X chromosome that will get inactivated. And it will be different in different cells, okay? So, a different X chromosome might be activated in the face versus the hand, or, it's not going to be the same. That's why there is some amount of random chance, if you're trying to clone somebody, right? If it's a female, the X chromosomes, in some cells, something will be silenced, in other cells, something else will be silenced. Right? So female mammals permanently inactivate one of the two X chromosomes by condensing it into heterochromatin early in embryonic development, right? And thereafter, in all of the many progeny of the cell, the condensed and inactive state of that X chromosome is propagated, right? And the rest of the interphase chromatin, which is in a variety of extended states. So we talked about heterochromatin. Okay, that is compact. That is the one which has got real compact packing. The other interphase chromatin, okay, which is not heterochromatin, it is called euchromatin, right? So again, this is terminology: heterochromatin and euchromatin. Good example of heterochromatin is in the X chromosome of female mammals. Okay, in every cell, one of the X chromosomes is inactivated by being condensed into a region of heterochromatin. Now, in a typical differentiated eukaryotic cell, some 10% of the euchromatin is in a state in which it is either actively being transcribed, that means it's producing messenger RNA, or is easily available for transcription. And this 10% of euchromatin is known as active chromatin and is the least condensed form of chromatin in the interphase chromosome. Now, by moving genes around in the genome, you can dramatically alter gene expression, right? So if a particular gene is moved from a region of heterochromatin, heterochromatin is transcriptionally inactive, very compact packing, right? If you move it from that region to a region of active chromatin or vice versa, its transcriptional activity is likely to be altered. For example, if you're looking at yeast cells, all right, there is a particular gene that is called ADE2, okay, which is responsible for which produces a protein that is needed in adenine synthesis. The normal location of that gene is here in the middle of the chromosome, right? If using artificial means you're able to move this guy to the telomere region, right, you're going to make this transcriptionally inactive. That means that particular yeast cell is not going to be able to produce adenine, right? The net result you see is that instead of this white colony of yeast cells, you're going to see a red colony here, okay? Because it cannot, once you move this guy over here, it cannot produce the appropriate protein, all right? And that causes this change in color, okay? Now, of course, you will see some white patches here because the silencing is not complete, right? You move this here, yeah, it's not transcriptionally active, but a little bit of it will get transcribed, right? So you can see this effect. Another example is if you look at fruit flies, okay, that's on the right, all right? Fruit flies, their eyes are red in color, okay? And there is a gene that's called a white gene. It's you know, in this figure, right, at the top, this is at the normal location, right? Now you can, I mean, today it's possible to do it artificially in the lab, okay? But you could move this thing, or even due to some random event, some rare chromosome inversion could take place. This thing could end up here, right? And it has come very close to heterochromatin, right? So it has become transcriptionally inactive, right? So originally you were having red eyes. Now you get a fruit flies that have this kind of mutation. They start having eyes that have red and white in them. Okay. So the point is that, you know, the gene expression, right, can be dramatically altered if you move the particular gene to a region of euchromatin, to a region of heterochromatin which is transcriptionally inactive, right? So you can do that. And it's possible to even produce more dramatic changes. Any questions? No, you can have something that will cut it and put it over there. You know. Yeah, we will talk about those techniques later on, right? Not in this chapter because, see, after we finish this first sequence of chapters, then we will talk about genetic variation which arises naturally in nature. And now today, in the last 40 years or so, we have techniques, okay, where you can take the DNA from one organism and, you know, cut it and put it in the cell of some other organism, you know, because the DNA from all organisms looks the same, right? So you can mix things up and, you know, try out all kinds of, weird experiments, right? So when I say moving, here of course it'll be something that happened purely by chance, some random event. Okay? And there are lots of things that go on in cells because of which this could happen. You know, you're doing cell division, right? When the DNA replicates, okay, it has to be separated out. Some mistake is made. Okay, one part breaks off from here, attaches over there, right? Things like that. If it somehow moves to the heterochromatin region, then it will become transcriptionally inactive, and you will see the effect at the observational level. All right. Any other questions? Yes, sir. No, see, like you, no, moving along DNA is different. You don't change the chromosome, okay? Like when DNA polymerase gets onto DNA, it is moving along the DNA, okay? But it's not damaging the DNA or doing anything. It's making copies from there. Here, it's like maybe you cut it, okay? You cut it with something, and then you stitch it back over here, right? There are ways of doing that. There are reagents that will do it for you, right? Maybe a cannot differentiate between DNA and chromosome. Because I understand that. No. Chromosome is basically the packaged DNA. So you have double-stranded DNA, right? Then, and if you look at the human genome, there are 30 billion nucleotides, okay? So there are 30 billion letters, and then you have the complementary base pairing, right? This is distributed between 23 pairs of chromosomes, 46 chromosomes, okay? So divide 30 billion by 46, that, I mean, assuming if they are more or less equal length, which they are not, okay? Then you can figure out what the length of each chromosome is going to be. Chromosome is basically just packaged DNA, that's all. Or organized DNA, okay? Yeah. But what I'm saying is that it's like you cut off a piece from here, okay? and then put it in here or something like that. Yeah, yeah. So now we get to the central question in the study of gene regulation. Okay, how does a cell specify which of its many thousands of genes should be expressed as proteins? Right? Because we have made the argument before that every cell from the same organism has exactly the same genome. Right? So how does the cell decide, which of these genes should be expressed? So this question is especially important for multicellular organisms because as the organism develops, many different cell types are created starting essentially from the same precursor cells. Okay. The single fertilized egg is going to give rise to the hair cells, the skin cells, the liver cells, the heart cells, the brain cells, everything. Okay. So how is that accomplished? Okay. Now this is called cell differentiation. Like initially you have a cell that's a stem cell, all right, embryonic stem cell. It differentiates means it's going to go in a particular direction. It could become a skin cell, liver cell, eye cell or whatever. Okay. So this differentiation happens because cells make and accumulate different sets of RNA and protein molecules. Right? That is what will decide what kind of cell it's going to become. Right? And to basically demonstrate that conclusively that every cell from the same organism is identical, there is an experiment that biologists did, right? So basically you have an adult frog here, right? You take an unfertilized frog egg, right? Take the egg, then treat it with ultraviolet light. Ultraviolet light can destroy DNA, right? I mean, you have all the thiamine dimers and all that. So you give a heavy dose, okay? Maybe induce double-stranded breaks, the nucleus is gone, okay? So this is now an egg which does not have a nucleus. So now you go and take a skin cell from this frog, right? From that skin cell, take the nucleus out, right? Put that in the egg, right? Okay, so you've taken the information from the skin, put it now back in the egg, okay? And now you fertilize it, all right, with a sperm. And then you see if that'll produce a fertilized egg and you will get a tadpole, right? It may not happen in every case, but even if in one case you get a tadpole, that's enough to show that the information in the skin cell, all right, has all the information that was there in the egg, okay? So people have done these kind of experiments, And again, I mean, a lot of times these are not successful because they have to be done under the right conditions and all that stuff, but they can show conclusively. So this means that every cell in this organism has got exactly the same DNA, okay? It's a question of which genes are turned on and turned off, okay? So interesting, right? I mean, and that is the reason, okay? Like you hear a lot of controversy about, stem cell research and things like that, okay? Because if you have embryonic stem cells, okay, from that you can grow any organ, okay? So like, like from your skin cell, they don't know how to do it, okay? There are people at some other schools like MIT and all that were trying to do that and to get stem cells from these because these are differentiated, okay? They're already differentiated. You cannot change it because imagine if you can grow nerve cells, all right? Like these days, see what happens is that nerve cells, they will last an entire lifetime without dividing. So that's why if somebody gets a spinal injury or has brain damage, then you're pretty much confined to a wheelchair or, you're immobilized, okay? You don't heal, all right? The cells in your liver, on the other hand, they are being replaced, they keep on regenerating all the time, right? Nerve cells do not, okay? So how would you go about, producing nerve cells for somebody that has been paralyzed? If you had access to that person's embryonic stem cells, okay, from that by turning on the right genes, all right, on and off, you'll probably be able to produce the nerve cells, okay, or heart, liver, or whatever, okay, instead of doing transplant from a different animal. So that's why these days, all right, I mean, maybe you guys are also not that lucky. I'm not that lucky, because, I mean, many years ago, all this technology was not there. But these days, I think the parents have the option, once the kid is born, okay, they, can go and freeze the stem cells from the umbilical cord, okay, and they have to pay like $70 a month or something like that. I remember my brother-in-law telling me. So later on when that kid becomes old, if they need like a new heart or liver or something, the possibility is there that they can go in there and produce it. In 50 years from now, maybe the technology will be there, okay, to do that. Because which today, you'll get somebody else's organ, it may work, it may not work, there might be rejection, there's risk of infection, all kinds of things, okay. But so the potential is very high, okay. But at the same time, you do need stem cells, okay. And that's why there is a lot of debate, about, because the only way they could get from stem cells is from, either umbilical cord or, aborted fetuses or things like that, okay. Like if it's a sibling or something, then somebody might have conceive and have another kid so that they can get those cells, like for treatment of leukemia and all that, But there are ethical issues over there. But the potential comes from this, from the fact that if it is a stem cell, right, embryonic stem cell, you can get it to differentiate to anything you want, right? Okay. Now, many of the proteins that are found in cells are found in all cell types. For example, the proteins that are needed to make DNA polymerase, RNA polymerase, ribosomes, etc. See, every cell needs to make its own DNA polymerase, right, for DNA replication. RNA polymerase for transcription, right, ribosomes for translation. So every cell is going to have the genes, right, or every cell is going to have these genes active, right. So these proteins which are needed for all the cellular activities, they are called, these generic proteins, they are called housekeeping proteins, right. And the genes that encode them are called housekeeping genes, right. At the same time, different cells will need different specialized proteins, right. And for example, hemoglobin in the blood is made only in reticulocytes, right? These are the cells which develop into red blood cells, because those are the only cells in the body that need to produce hemoglobin, okay? Your hemoglobin has got no function in your liver cells, in your skin cells and so on, okay? Only the red blood cells have to make it. So the gene corresponding to hemoglobin should be turned on, okay? Or should be producing messenger RNA only in the red blood cells, right? Similarly, the beta cells in the pancreas, those are the only cells in the body, right, that need to produce insulin. So the gene corresponding to this protein insulin should be turned on only in those, okay. Now, how does the cell go about controlling the production of protein? A cell can control the proteins it makes in a number of different ways. First of all, the cell can control when and how often a given gene is transcribed. That means when and how often a given gene is copied into messenger RNA. Okay, it can control that. Then the cell could also control how the primary transcript is spliced or otherwise processed. Remember, I said that in the case of the eukaryotic cells, right, you have coding regions, exons, right, interspersed with introns, right? And then splicing is the procedure by which you get rid of the introns, okay? And there is alternative splicing, like, if you have 3 or more of these exons, all right, then you could do it in different ways and get different proteins. So a cell could control how the primary transcript is spliced or otherwise processed. Then the third thing that the cell could do, it could decide, once it's been transcribed into messenger RNA, all right, to get protein, you still need to translate it. So the cell could make decisions about which of the messenger RNAs should be translated by the ribosomes. Because if you just do transcription, you don't do translation, you don't have any protein, right? So you haven't produced any protein. And the last thing that the cell could do is it could selectively activate or inactivate proteins once they are produced. Okay, which one do you think that the cell will use the most among these 4? Which one is the most efficient? Huh? Go. What? First one, yeah, because why waste your time, go through all of this and then inactivate proteins, So the one that the cell will do most efficiently is to try to control when and how often a given gene is transcribed, right? Because it will depend on the needs of the moment, right? If some protein is needed, then and only then it will be transcribed, right? It's not going to go through this whole thing, produce it, and then destroy the protein because it doesn't need it, right? At least in normal cells, that should not happen. So how is transcription controlled? So we agreed that control of transcription is probably the best way to control the amount of protein in the cell, right? Now transcription is controlled by proteins binding to regulatory DNA sequences, right? So it is true that you have this region that codes for a protein. RNA polymerase gets on and starts the transcription, but there are other factors that have to kick in. Okay, there is other stuff that controls. So the promoter region of a gene, that's the portion that precedes the place where the transcription is going to start. So the promoter is the region where the RNA polymerase latches onto the gene. So the promoter region of a gene attracts the enzyme RNA polymerase and correctly orients it to begin its task of making an RNA copy of the genes. The promoters include the initiation site where the transcription is supposed to start, and a sequence of about 50 nucleotides that extends upstream from the initiation site, right? This region contains sites that are required for the RNA polymerase to bind to the promoter. Now, in addition to that, there are what are called regulatory DNA sequences. that are needed in order to switch the gene on or off. Right? Just putting RNA polymerase over there is not sufficient. There is some other stuff that is required. Some of these regulatory DNA sequences are as short as 10 nucleotide pairs and act as simple gene switches which will respond to a signal, right? And this occurs primarily in bacteria, and we'll see an example of that. Other regulatory sequences could be very long, as many as 10,000 base pairs, and they could act as molecular microprocessors, all right, taking in several inputs to determine the transcription rate. And this occurs in eukaryotic cells, like in human cells, okay? So it's not like we invented double E's invented microprocessors or something. You know, inside the cell also there is, a lot of decision making that goes on, right? A lot of information is collected, and then decisions are made about whether the cell should divide or not, or a particular protein should be produced or not. Now, so in the promoter region, you have these regulatory DNA sequences. Okay, these sequences don't act by themselves, but they have to be bound by gene regulatory proteins, right, which will recognize them. Now, gene regulatory proteins can either suppress transcription, right? They can shut it off, or they can enhance transcription, right? So activators will turn genes on, and again, a gene is turning on means transcription is starting, right? DNA, corresponding stretch of DNA is being copied into messenger RNA, right? Off means transcription is shutting off, right? So repressors would turn genes off. Now this is a good example, okay, of how a bacterium, right, can turn genes on and off in response to its needs, okay? And this is called what is called a tryptophan operon. Tryptophan is one of those 20 amino acids, okay, that the bacterium needs. It's one of the 20 amino acids that are present in nature. This operon is also very similar to what I referred to last time as the lactose operon, or the lac operon. Remember, I said that in response to the needs of the moment, the bacteria can switch from E. coli can switch from not producing the enzyme that is needed to metabolize lactose to producing the enzyme that is needed to metabolize lactose, right? It's the same kind of thing happening here, okay? Now here, see, these guys, E, D, C, B, and A, these are the genes, okay, that code for the proteins that are needed, or the enzymes that are needed for synthesizing tryptophan. Tryptophan is one of the amino acids, okay? So in a bacterial cell, we have mentioned that before, that you could have many different proteins that could be transcribed from a single messenger, transcribed into a single messenger RNA, right? Because you could have many genes, right, that are transcribed together. So this is an example of that. So this is the E. coli chromosome, and you have A, B, C, D, E. These are the DNA sequences that actually will produce this messenger RNA, right? And then when you translate these, you are getting the different enzymes that are needed for synthesizing the amino acid tryptophan, right? Now RNA polymerase has to get onto this promoter region and start this transcription, okay? Now, so the operon is basically several genes which are transcribed as a single mRNA. That happens in the case of bacteria. If you're looking at eukaryotic cells, it's like one gene will result in one mRNA. Okay? You're not going to have several proteins coded in the same mRNA. Now inside this promoter region, there is also a DNA regulatory sequence. Now when a repressor molecule binds to this sequence, what happens is that RNA polymerase cannot get on here, okay, and the transcription will stop, okay. However, the repressor molecule can come and bind this regulatory sequence only after it has bound several molecules of tryptophan. So if you have a lot of tryptophan in the cell, some of those molecules will bind to that repressor, right? That repressor will get in here. It won't let RNA polymerase do any transcription. So the tryptophan synthesis will stop, okay? So this is like the float in your toilet flush, okay? I mean, if the water level is too high, then the water is cut off, right? I mean, you flush again, so it's exactly the same kind of thing. It's like a feedback mechanism, right? So if the level of tryptophan is high, then the repressor molecule is bound by some tryptophan. The shape changes. This is an allosteric protein, right? Remember, we talked about that. Maybe by now you've forgotten what an allosteric protein is. It has two binding sites. On one side you put something in, the shape changes, the behavior at the other side is altered. So it's the same thing. If you have this repressor molecule is an allosteric protein, because if it's bound by tryptophan, then its shape changes, okay? Once it's bound by tryptophan, its shape changes, then it latches onto this gene regulatory sequence. RNA polymerase cannot get on, tryptophan synthesis stops, right? Then when the level of tryptophan in the cell drops, all right, the repressor molecule is not bound by tryptophan anymore, right? So then it cannot latch onto this gene regulatory sequence. It moves off. RNA polymerase gets on and does the transcription. Right. So, so this is exactly the same thing like the lactose operon. Okay, except that now it's tryptophan synthesis. Over there, it will be the enzymes that are needed for metabolizing lactose. Right. And that was discovered in the 1960s. That's why you can see this is something like a Boolean type of logic thing. You know, if that thing is not there, okay, then go and produce it. Okay, if it is there, then shut it off. Shut off production. Right. So this was discovered by Jacob and Menard in the 1960s, and they did get a Nobel Prize for it. So in biology, I don't know whether it's good or not, but you know, the thing to notice, what is remarkable is that everybody that discovered a small piece of that puzzle, right? These are of such far-reaching consequences that all of them got Nobel Prize. Like the guy who figured out, right, that the genetic code is triplet, that guy got a Nobel Prize, right? So if you're looking for a Nobel Prize, you know, this is the wrong field that you're in. Okay. Engineering is probably not the one. You know, like I mean, there has been one guy who was an engineer who got the Nobel Prize for physics. Do you know who that was? Or he was associated with Texas A&M, right? Who? Yes, Jack Kilby. Yeah, yeah. He used to be a distinguished professor here. He has the patent on the first integrated circuit. Okay, so and that is a far-reaching consequence because you know you name it, your computers, everything is using those. Okay, so he shared a Nobel Prize in physics. Okay, but other than that, I don't know of too many engineers that got Nobel Prizes. You know, so you're in the wrong field. Yeah, you're doing applications and all that. You don't get the so one prize you've already lost. Okay, so one less prize to work towards. Huh? So as I said, when the level of tryptophan in the cell is high, the transcription of the operon will stop. On the other hand, when the level of tryptophan in the cell falls, the repressor is no longer able to bind the regulatory sequence, and so RNA polymerase can start transcription of the operon so that more tryptophan can be produced. Right? The tryptophan repressor protein is always present in the cell, although at a low level. Right? It's not at a high enough level. So such unregulated gene expression, and this is again biological terminology, is called constitutive gene expression. As opposed to induced gene expression is when a protein is produced in response to a stimulus. And again, for those of you that are working in the center that are going to, these are things that you're going to see over and over again. Okay, I'm covering all this in the course so that every time you read a new paper, you're not thrown off. Okay, like they talk about it, the biologists and all, like, you know, ABCD, ligand protein interaction. You need to know that terminology. You cannot be substrate, okay? Then, you know, the different sites and all, and binding site and all that stuff. That's terminology that you need to know. And even those blots that we talked about, Western blot, Southern blot, and whatever, not, it's like the ABCD, okay? It's like if you were a double E, if you didn't know what a capacitor is or inductance or, you know, something threw you off, or, you know, time constant threw you off, then you cannot be a double E. So it's the same thing. You need to know all of these things, you know, if you're going to be working at the interface of biology and engineering. Now, just like you have repressors, you can also have activator proteins where, for example, you know, there is some binding site here. If the activator protein doesn't bind here, okay, then RNA polymerase, this promoter region is not fully functional. RNA polymerase cannot go and synthesize the messenger. It cannot go ahead and do the transcription and produce the necessary messenger RNA. However, when this site, when this DNA sequence is bound by this activator protein, then the promoter becomes fully functional, and then this one can go and produce the messenger RNA. So just like you have repressors, that tryptophan operon was an example of a repressor, you can also have activators also. Okay, and in fact, you know, for example, I mentioned some time ago about cyclic AMP. Does anybody remember what that was? Cyclic AMP. That is a signaling molecule, you know, that can activate things. Yeah, there was a cycle from the 5' to the 3'. Because when you see cyclic AMP, that is a low energy signal, right? So that can signal to the cellular machinery that, hey, you need to start transcribing genes that are going to break down sugars. Okay. So that's a good example of, in fact, I think it's called a catabolic activator protein or something like that. You know, that will, when it's bound by cyclic AMP, the catabolic activator protein becomes active, and it will start the transcription of all those genes that are needed for breaking down glucose and producing more energy. Now, what I showed you about that single repressor or that single activator, that is usually true in bacteria, in E. coli where it's easy to study, okay, in bacteria and simple cells. In the case of eukaryotes, the transcription is a lot more complicated, okay. It's as though like, it's not like just one molecule, it's like a big committee that has to come together, right, and then make the decision about whether the transcription will take place or not. Right. So we next discuss key differences between initiation of transcription in prokaryotic cells and eukaryotic cells. First of all, prokaryotic cells contain a single type of RNA polymerase, while eukaryotic cells have three different RNA polymerases that are called RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Right? RNA polymerases 1 and 3, they will transcribe the genes that encode transfer RNA. You all know what that is. Okay, those are those adapter molecules. Ribosomal RNA, those are the RNAs from which the ribosomes are made. And small RNAs, right? Like they call them microRNAs, and they have some special function in the cell. On the other hand, RNA polymerase 2 is the one that transcribes the vast majority of eukaryotic genes, including all those that encode proteins. That means all those genes that produce messenger RNA. Okay, that's one important difference. So there are three types of RNA polymerase. Second thing is that in the case of eukaryotic cells, that RNA polymerase 2, it cannot initiate transcription on its own. Okay, it needs a lot of friends. To initiate transcription, eukaryotic RNA polymerase requires the help of a large set of proteins that are called transcription factors. And if all those guys are not there, it's just too bad. RNA polymerase can be there, but transcription will not take place. So these guys, these transcription factors, again, this is also terminology that you need to know. These are thought to position the RNA polymerase correctly at the promoter to aid in pulling apart the two strands of DNA, because you have to have a DNA template exposed. Okay, you cannot work with double-stranded DNA, right? Then to release RNA polymerase from the promoter once transcription begins. So all that is enabled by these transcription factors. The assembly of transcription factors and their role in initiating transcription at the appropriate location are shown in the next slide. And again, this is a schematic diagram. So this is like the promoter region. It has a box that is called a TA, Tata box they call it, okay, because it's TA, right? Thymine, adenine, thymine, adenine. So there is some consensus sequence, right? It's not a unique sequence, some consensus sequence that will determine the start of transcription, right? And yeah.
Tata box, is it always there?
I think so, yeah. But again, I mean, check. I mean, don't hold me to it. I have seen that thing, So the Tata box is there, yeah. That's one of the indicators. But again, I mean, the sequence over here, it'll be some consensus sequence, okay? It doesn't have to be just one particular sequence, right? And you guys are experts in stochastic processes, so you can do the probability of the transcription taking place given what you get and all that stuff, and all that good stuff, probability of A given B, B given A, and Bayes theorem and all that, and you can do fun stuff with it. But once this is there, transcription has to start here. So then these guys are all the transcription factors, and there are many of them, like TBAF and all that. I don't even know what the detailed structure is, but all these things will come together. They will form a complex like this. See, everything is assembling. They form a complex. These guys are all transcription factors. Then there is some transcription factor, one of these, okay, maybe TF2H or something. It has got protein kinase activity, so it phosphorylates the RNA polymerase, right? Add a phosphate group, right? If it's a kinase, it'll add a phosphate group. Immediately, the result of this is that all these friends that came to help, right? They helped get it started, right? But now it doesn't need its friends anymore. Okay, your friends help you get started. Now you just want to run on your own. Okay, get rid of all your friends and run. Okay, so that's what is going to happen. It'll get rid of these transcription factors, and then the transcription starts. Okay, there's the RNA that is being produced. Okay, so it is a lot more complex. Not only that, on the next slide, I'm going to show you that it doesn't even have to be all these things that are binding here. There could be something that is binding thousands of nucleotides away, right? That can have an effect. That can come and link up here and be one of the transcription factors. Okay, it could be 10,000 bases upstream, downstream, right? So this is like a committee. If all these guys work together, then the transcription will take place. Okay, one of these guys gets messed up, you might have transcription at the wrong time. Okay, and it could lead to diseases like cancer and so on. So once the initiation complex has assembled, the general transcription factor TF2H, which contains a protein kinase enzyme as one of its subunits, will phosphorylate the RNA polymerase. And this phosphorylation is thought to help the polymerase. And again, I'm saying this is thought to because nobody knows exactly for sure. Okay, but this is all, maybe some of it is known since I read that book, but if they would have said for sure that it helps the polymerase. They wouldn't say it is thought to help the polymerase. All right. So disengage from the cluster of transcription factors, allowing transcription to begin. then this picture shows you that in eukaryotic cells, the gene regulatory proteins can influence the initiation of transcription even when they are bound to DNA thousands of nucleotide pairs away from the promoter. So this is the promoter, and then you have a lot of stuff in between. There's some activator protein that got bound here, right? But then this is linking up to this set of transcription factors, and it can affect the initiation of transcription. And a good example of a transcription factor is a gene that is called P53, right? You may have heard of that. It is implicated in many human cancers, okay? Like maybe a large percentage of the cancers have a mutated P53 gene. Now finally, eukaryotic transcription also must take into account the packing of DNA into nucleosomes, right? Which is what we discussed at the beginning of this class, and more compact forms of chromatin structure. And because of this compact packing, if the DNA is in the nucleosome, the transcription factors cannot assemble. There is not enough room, right? So the start of transcription will have to be from some place other than a nucleosome. It may have to be opened up, right? But then once the transcription is started, then those factors are gone, okay? And then RNA polymerase can still keep on transcribing, right? So it is believed that the nucleosomes prevent the general transcription factors of RNA polymerase from assembling on the DNA and can therefore hinder the initiation of eukaryotic transcription. Now, most bacterial genes are controlled by a single activator or repressor protein, just like I gave you the example of that tryptophan synthesis, that repressor protein. Okay. On the other hand, most eukaryotic gene regulatory proteins will work as part of a committee of regulatory proteins, all of which are necessary to express the gene in the right cell in response to the right conditions at the right time and at the required level. Right? And this is referred to as combinatorial control, and it is very similar to Boolean logic, familiar to most engineers. Right? Like if you have A and B and C and D, right? Everything has to come in with one before you get the output that you want, right? So here also it's like a committee. So indeed, using Boolean functions to model relationships between expressed genes is a theme that we will encounter in the later part of this course. Okay, when we make the transition from molecular biology to basically genomic signal processing or engineering applications or informatics. Now, in the case of eukaryotic cells, combinatorial control of gene expression can create different cell types. For example, fibroblasts, which is that's one class of cells which start from the same class of embryonic stem cells as muscle cells. You can convert fibroblasts into muscles or into myoblasts by just expressing the gene myosin D, right? So it appears that fibroblasts, which are derived from the same broad class of embryonic cells as muscle cells, they have already accumulated all the other necessary gene regulatory proteins required for the combinatorial control of the muscle-specific genes. And just addition of this one gene, myosin D, the protein, completes the unique combination that directs the cells to become muscle. So it's like an AND gate. If you have 50 inputs, okay, all the others are 1, okay. You put in 1 in the last input, and you will have a 1 at the output, right? Similar to that. Now consider the hypothetical example here, and I mean this is just a toy example. Okay, let's say you have 3 genes. Okay, they can either be expressed, right, which will be 1 or 0, right? So there is a total of 8 possibilities, right? So when this cell divides, you can go ahead and change one of them to 1. Okay, you can express that gene, right? Then again let these guys divide and again change, maybe the second one and so on. So if you do that like after 3 rounds, you have 8 different possible cell types. Okay, starting from that single cell over there. Because this guy has got 1 gene expressed, that guy has got 2 other genes expressed and so on. Okay. Any questions? Now some highly specialized cells never divide again once they have differentiated. For example, nerve cells or neurons, they never divide. But there are many other differentiated cells such as liver cells which will divide many times in the life of an individual. All these cell types, however, give rise only to cells like themselves when they divide. This means the changes in the gene expression that give rise to differentiated cell must be stored in memory, and they must be passed on to its progeny through all the subsequent cell divisions. Right? And cells have several ways of accomplishing this. One possible mechanism involves a positive feedback loop, where a key gene regulatory protein is going to activate transcription of its own gene. Right? So more of that protein is going to be produced, and when the cell divides, it will make sure that both the copies have enough amounts of that protein to get the characteristics of that particular type. Right? In another mechanism, the condensed chromatin structure is faithfully propagated from parent to daughter cell, even though DNA replication intervenes. Right? An example of that is the inactivation of one X chromosome in female mammals. Okay? Because the progeny from that particular let's say the paternal chromosome is inactivated, then all the descendants from that will have the paternal chromosome inactivated. Although in between there is DNA replication before this, every time the cell divides. Right? Now, this is a very interesting example, all right? So this is an experiment to demonstrate that alteration of the expression status of a single gene can trigger the development of an entire organ, and that too at an abnormal location. So now there's a gene called EY in flies, okay, or eye, and it's called PAC6 in vertebrates. It's crucial for eye development. In this experiment involving fruit flies, the gene EY is expressed early in development using artificial means. Okay, and today we have the last 40 years we have the technology to do that in cells that normally go to form legs. In fact, even in this center there is technology to do this kind of manipulation. Like Mike Bittner, some of you may be familiar with him. He has the techniques to take cancer cell lines and put in different mutations and things like that. Okay, so you guys have come to the right place. Okay, it's not just theory and attending a class like this. We have the ability to do this kind of modification, and not only that, generate time course data by following gene expression activity over time. Okay, without killing cells. Okay, we can do that. So the result is that in this case, the corresponding fruit flies, they develop eyes in the middle of the legs, right? So thus this experiment shows that the action of just one gene regulatory protein can turn on a cascade of gene regulatory proteins whose actions can result in the formation of an organized group of many different types of cells. Okay, you're getting a complete eye on the leg. And you could probably try it somewhere else too, if you wanted. Okay, that finishes this chapter and we'll stop here for today. Next chapter, I will talk about genetic variation, right? How, life on this planet, the variety of life you see is because of alterations in the DNA over evolutionary time. Then the chapter after that is when we'll talk about making changes in the DNA in the lab, okay, which is basically what you guys are interested in, or why you came here, okay, because this stuff is happening in nature, okay. I mean, why would you come to study this stuff? It's going to happen by itself anyway. All right. So any questions? So let's stop here for today.